Okay, folks, so I don't want to go over the warm-up today because I want you all to have time at the end of class to start working on some of your missing assignments, as I think almost everybody in this class has missing assignments. Give me 1 second so I can do something. Oh. The internet is just really slow in this building, so I apologize. Yes. It's loading so slowly. All right, so I'll just go ahead and start from here. Today is February 26, 2021. It is the third lesson of our 5th unit. And in this lesson, we're going to go over the first part of transcription. So this lesson will be continued on Monday as well. And this is an extremely important process, so I want to make sure that you all are taking accurate notes. So we will take our time. There's no reason to rush. We don't have an assignment to get to today. Please do make sure that you're starting your note-taking process now. All of my people who are here in person have their notebooks out. And so yes, your note-taking process should begin now. The objective for today's lesson is to explain how DNA and RNA code for proteins and determine traits. Not only will we continue this lesson on Monday, but we will also introduce translation, which is the vital second part of this process, on Tuesday. So we still have a couple of days after today to focus on this same objective. The essential question today: how does genetic information in the nuclei of eukaryotic cells get to their ribosomes, which are in the cytoplasm? So this process requires some amount of travel through the cell. Of course, the DNA in a eukaryotic cell is found in the nucleus. And so in order to get to the ribosomes, that message has to be transferred. All right. Let's go writing. No, we're good. Okay, I'm going to start off with the quiz today about DNA replication, which we talked about yesterday. All right, so the game code should be visible on your screen, but I am going to send a link to the quizzes now. So you can just click on that link, or you can go to joinmyquiz.com and enter the game code 532926. Thank you, Jason, Pedro, Naftali, Brian, Breon. Thank you all. We've got 17 people in class today. And I think a couple more who are in person, who are not actually in this call. So we should have at least, I'm going to say at least 15 folks, guys, in this chat. Let's get 8 more. Thank you, Gloria and Rudy and Kadaria. And thanks for being here, Brianna. I will change my attendance accordingly. Okay, I'm going to wait for 5 more people. Can I get 5 more people to join this call? I know some people are probably dealing with slow internet, just like me. Okay, thank you, Michaela, for letting me know. 5 more people to join this game. Thank you, Abraham. 4 more people. Can I get 3 more people? Chandler. Oh, your computer's not working. Can you join from your phone? Who else hasn't joined yet? Kathy. Oh, there's Kathy. Sorry, Kathy. I see your name now. There's Jason. Cindy, can you join this game? The link is in the chat. Send it one more time. All right, we're going to go ahead and get started. I'd love to see a few more of you join. You can obviously join while the game is in process or progress, rather. You guys know where I want to be. I want to see that accuracy up as close to 70% as possible. All right, so we're starting to have people finish up. The accuracy is actually it's at a pretty good spot, guys. So 63% is not 70% where I would like to be, but at this point in the unit, this is not bad. We're going to try some innovative things next week, which I'll talk about a little bit later in the lesson today. And I think that this will really benefit those of you who are needing some help at this point. Of course, keep in mind that I expect our midterm will be a week from Monday. So we've got 9 days essentially before the midterm exam, and we're going to need to make the most of our time in order for everybody to do well. We still have some folks who are finishing up, I see. And that's okay. So I'm going to take about 2 more minutes to allow folks to finish up, and then we'll jump into the actual lesson for the day. We'll come back and we will review these questions later on. Okay, about 1 more minute, 1 more minute. You don't need to be done. You can keep working, but in 1 minute, we're going to switch back over to the actual lesson. Okay. Switch over here. Next. All right. So the central dogma of biology, what does this mean? The central dogma of anything means that it is the most important idea that you need to know about it. So the central dogma of biology is like the central most important idea of biology. And basically it states genes, which are obviously made of DNA, code for the proteins that we make. Your genes are the instructions that tell your cells which proteins to make. This is the central dogma, the most important idea in the study of life. Of course, we could take it a step further and say that those proteins eventually give you the traits, or give any organism the traits that make them what they are. So the process is there. Today, we're going to talk about transcription, which is the very first step, going from DNA to mRNA. You don't have your notebook, Naftali? DNA going to mRNA is the process of transcription. On Tuesday, we'll talk about the process of going from mRNA to amino acids. That's called translation. Those amino acids eventually become proteins, and the proteins give you your traits. So this is the most important idea in biology. All right, I asked these same two questions yesterday, but I just want to make sure that folks were paying attention and that they wrote down the answers. Keep in mind that when you see things that are highlighted, that's an indication that you should be writing them down. So the first question I asked was, why does our DNA need to be replicated? Who can tell me that? Why does our DNA need to be replicated? And I love what I can see Chandler and Pedro, who are both in the class, looking back in their notes. This is what I should be able to see you all doing if you were all here. Does anybody remember, why does DNA need to be replicated? Did you find anything, Chandler? Did you find anything, Pedro? Good, yeah. So Pedro says that it needs to be available for the daughter cells, AKA the new cells. So this is correct. Before the cell can actually divide, we have to have enough copies of the DNA to go into the daughter cell. So again, if you didn't have this answer already written down, we see that it is highlighted, so we should go ahead and copy it into our notes. Before a cell can divide during M phase, there must be a copy of the DNA for new cells. And while some folks are still writing, I know some of you probably have your hands free and can type into the chat. What is M phase? What does M phase mean? Or you can just unmute yourself. Brian, you need to go to the meeting. Give me 1 second. All right, I'm going to work through for you. Yes, please. Thank you. All right, so I asked a question. I said, what is M phase? What does M phase stand for? Oh, thank you, Rudy. I'm sorry, I didn't. Yeah, meiosis or mitosis. Good. And these are the two processes of cell division. Over the next week, we're going to do a lot of review just to prepare, I'm sorry, for the midterm exam. Okay, this was the second question that I asked yesterday. Where does DNA replication occur? The previous part, yeah, over here, yes. M phase, yeah. M phase stand. I can write it on the board since you don't have a Chromebook in front. Just wait for a second, Brian, until the escort gets here. Yeah, sounds correct. Yeah. All right, so end phase equals mitosis or okay, so can you see that? Can they see that? Yeah, they can see that. While we're at it, we might as well kind of talk about the differences between the two. Alex is here. All right. Thank you for joining us, Alex. All right. So, does anybody remember any qualities of mitosis? Some of the differences between mitosis and meiosis? Okay. Have a good weekend. Does anybody remember any of the differences between mitosis and meiosis? For example, I'll start you off. Mitosis produces 2 cells, whereas meiosis produces 4. That's good to know. Anything anybody else can give me, and feel free to take yourself off of mute just so we don't have to sit and wait for you to type. What are some other qualities of these processes that we can remember? Nothing? Okay. What about the fact that mitosis produces identical cells, whereas meiosis produces unique cells? What else do we want to mention? So meiosis produces what are called gametes. These are our sex cells, the sperm and the egg. Whereas mitosis produces what are called somatic cells. These are all the other cells in your body. And one other thing that I will mention, meiosis undergoes 2 rounds of cell division. So prophase 1, metaphase 1, anaphase 1, telophase 1. And then we do it again, prophase 2, metaphase 2, anaphase 2, telophase 2. Mitosis, on the other hand, only does each thing twice. So these are both types of cell division. I guess I should have included that at the beginning. They're both types of cell division, but in order for meiosis to produce 4 daughter cells, it needs to divide twice. Yeah, add one more. Okay, so that was some review from couple of units ago, but it's good that we can keep coming back to it. We're going to do a lot more review next week. The answer to the second question, which was where does DNA replication happen? Where we find DNA is where we would expect to have DNA replication occurring. This means that it occurs in the cytoplasm of prokaryotic cells because prokaryotic cells do not have nuclei, and it occurs in the nuclei of eukaryotic cells. You writing this? Okay. All right. This is a new question that I'm asking specifically for today's lesson. But what is a cell's primary purpose? I'm going to go ahead and give the answer for this because we don't have much time. The primary purpose of a cell is to make proteins. Proteins are absolutely essential to life because they complete a wide variety of functions for living organisms. They provide protection, they help to break things down, they help to synthesize new things, they help to carry and transport things, they help for communication purposes. So a cell exists, most cells exist to make some type of protein. And remember, the instructions to make a protein come from the DNA. The DNA carries the instructions to make proteins. Why don't you like pull up Canvas or something and work on one of the assignments? I think that's what you're doing right now? Okay. Do you need any help? All right. Ooh. All right, so we talked about these 5 examples of proteins way back in Unit 1. We talked about antibodies and enzymes, insulin, hemoglobin, and hormones. Does anybody remember what an antibody does? This has been something that has obviously been in the news a lot recently, but what do antibodies do? Who's coming to school next week? Breon, Ayana, Ardesia, and Alex. Does anybody know what an antibody does? Okay. Is anybody alive at home right now? Thank you, Rudy. They provide some immune functions. They help our body to fight off pathogens. A pathogen is a virus, bacteria, even poisons and other toxins are pathogens. So we need antibodies to protect us against them if we come into contact with them. We should all know what enzymes do, but we talked about this, pretty in depth in Unit 1 as well. Enzymes speed up chemical reactions. So most chemical reactions that naturally take place in our bodies would be pretty slow without the help of enzymes. So we need the enzymes to help us do them faster. Insulin is probably a protein with which you all are familiar because we know that it has something to do with diabetes, but insulin is actually there to help maintain or regulate levels of glucose in the body. So if your sugar levels are too high, your body produces insulin, which then signals another protein to break the sugar down. People who are not able to produce insulin, either because they were born with a mutation leading to type 1 diabetes or because they developed it as a result of their lifestyle over the course of a number of years, leading to type 2 diabetes, those people are not producing insulin, and so their body does not break down sugar very well. This becomes dangerous because we know of this concept called homeostasis. We need to be able to regulate different chemical properties in our bodies: sugar, temperature, pH, oxygen, carbon dioxide. All of these things need to be balanced. And so we rely on proteins like insulin to help us maintain that balance. Then there's hemoglobin, and there are also hormones. Hemoglobin carries oxygen in our blood, and then hormones serve as chemical messengers between cells. They help the cells in your kidney, for example, communicate with your brain, or the cells in your brain communicate with your reproductive system. Okay. So we know that there are two types of nucleic acids, two general types. There's DNA, and then there is RNA, and there are clear differences between the two. So I want you guys to take a look at this slide and tell me if you can notice any differences immediately. And tell me what those differences are. What do we notice? Can someone tell me just one difference? Just give me one difference. I know it's Friday. Thank you, Jason. One is stranded. They're both stranded. What do you mean by that? They're differently stranded, though. You are right. That is a difference, but can you tell me more about it? Okay, yeah, the RNA is single stranded, whereas the DNA is double stranded. There are two strands that are connected to one another in the middle and wrap around one another. What else? What other differences do we notice? There's another pretty obvious difference here. Maybe if we focus on the colors, we'll see. Okay, nothing. You guys are. Thank you, Brian. The RNA doesn't go in a pattern. Okay, I'm not exactly sure I understand what you're saying there. Let's take a look, and I want you all to write this down. I cannot emphasize this enough. Even when you write things down, guys, there's research that tells you that your brain learns it better when you actually write it down. And this is not something I'm making up just to serve my agenda. Your brain actually is set up so that when you write things down, it helps you to memorize it. So we should be writing these things down. There's no reason why we shouldn't have, many pages of notes at this point in the semester. So here's what I want you to write. DNA stands for deoxyribonucleic acid. That is a long word, but it includes some important information, including the fact that DNA is a nucleic acid. RNA, on the other hand, stands for ribonucleic acid. Jason correctly identified that DNA is double-stranded, whereas, as he put it, the RNA is alone. RNA is single-stranded. DNA has a sugar called deoxyribose sugar, whereas RNA uses what's called just regular ribose sugar. And then DNA has the nitrogen bases A, T, C, G, whereas RNA has the nitrogen bases A, U, C, G. So please make sure you've got this chart copied down. I see your message, Sade. I will do that. We've got 20 minutes left. And then that last row says that there's only one type of DNA. That's true. But there are several types of RNA that come up at different parts of the processes that we'll talk about today and on Monday and on Tuesday. Okay. So here are the different types of RNA. There is mRNA, which carries the DNA message. It stands for messenger RNA. You should be writing this down as well. mRNA carries the DNA message. rRNA is a part of the ribosome. The ribosome is made up of some proteins, but also some RNA called ribosomal RNA. And then lastly, there is transfer RNA, tRNA. And this is what actually brings the amino acids to the ribosome. It transfers the amino acids to the ribosome so that they can be linked together to start to make a protein. So please make sure that you're writing down these three types of RNA. Okay. We don't need to watch this right now. Interesting, but don't need to watch it. Okay, so transcription, of course, is this first part of protein synthesis. The goal of transcription is highlighted in yellow. That means you should write it down. The goal of transcription is to convert DNA into an mRNA message. The location of transcription, of course, depends on the type of cell. So transcription occurs where DNA is located. In a eukaryotic cell, DNA is located in the nucleus. In a prokaryotic cell, DNA is located in the cytoplasm. We're going to talk more about this other process of translation, but we should always remember that transcription comes first. The way that we can remember this is that transcription is alphabetically first, because C is alphabetically before L. So transcription comes first. Next slide. Transcription is a relatively complicated process, but we're going to just summarize it with these 3 quick steps. First, the DNA double helix is unzipped by an enzyme, just like how we unzip it for DNA replication. Then there's another enzyme that comes in called RNA polymerase. Should sound familiar because we talked about DNA polymerase yesterday. But RNA polymerase uses the DNA template to make an mRNA complementary strand. And then the mRNA is going to leave the nucleus and travel into the cytoplasm. All right. So just to create an analogy, it's kind of like decoding a text message. It's still written in the same language, but it's just written a little bit differently. So DNA and RNA are both written in the same language. They both use the language of nucleotides, A's and T's and C's and G's. They have sugar backbones, a sugar phosphate backbone, so same language. But of course, RNA uses U's, whereas DNA uses T's. And RNA uses a ribose sugar, whereas DNA uses deoxyribose sugar. So same language, but just written a little bit differently. Kind of like in a text message, if you were to have abbreviations, the abbreviations are there in English for the most part, but you might need to explain them a little bit differently. All right, so in your notes, why don't you quickly copy down these 2 DNA strands, and then tell me if you can get the correct RNA strand that gets transcribed from them. So I'm not going to do anything. I want to give you guys some time to write this down and work on this by yourself. In the meantime, I'm going to copy my own onto the board. So remember, there are no T's in RNA. There are only U's. Does anybody think that they know what the correct mRNA transcript is here? You think you got it, Pedro? Good. Excellent. All right, so I'm going to scoot this over so you guys can see. But Pedro did just get this correct. So what we're doing here is an mRNA transfer from this DNA strand. Go to the next. So the A gets transcribed into a U. T gets transcribed into an A. A gets transcribed into a U. C gets transcribed into a G. G gets transcribed into a C. So now that you guys have seen it done correctly and you've written it in your notes, hopefully, do the next one as well by yourself. Make sure that next one is also written in your notes. You think you could transcribe that? Okay. What are you thinking about? This very first. Okay, so keep in mind when we're transcribing something, Pedro, what does A become? Yeah. What does T become? What does C become? And then what about G? Good. Okay, so this is what we mean by transcribing something. Going from one letter to the other. In this case, A wants Q. C wants A. C wants G. C wants C. So down here, T is going to come as well. T, A. Good. Good. So now you got a handle, right? Okay, cool. Now this is only for the process of transcription.
So like that's the process. So like any how much transcription?
Yeah, anytime you're transcribing, it doesn't matter. This could be 3,000 letters long, and you can still be able to follow the same. Yeah.
So will the layers always be the same? Or will they do different layers with different transcription?
There are only these same 5 letters, A, T, C, G, and U. Notice that DNA has T, whereas RNA has U. So there are no U's in DNA, and there are no T's in RNA. Okay, so same 5 letters, it's not like you're going to randomly throw in an L or an S. We're going to be using these same letters. All right, can somebody at home, now that my people here have figured it out, can somebody at home complete this transfer for me? We left off with A, G, C. What should come after that? What should the G become? No, G does not become U. G does not become T. G does not become A. Guys, G, look, can you see the top of the board? Yeah. G becomes C. Thank you, Gloria. And then what comes after that? What does A become? There are no T's here. We only have U's. This is RNA now. Look at the screen. RNA uses a U instead of a T. What does T become? Thank you. A. Now it's even closer. Hopefully you guys can see. ACC, what should I have? What should it be transcribed to? Yeah, U, GG. Thank you. And then the last one, AG. Yeah, that should be UC. All right, so the takeaway here, RNA does not have T's. But so A is going to become a U. T still becomes an A, but A becomes U. All right, so please make sure you are reviewing and getting assignments completed this weekend, but also take some time to relax. I'll talk to you guys on Monday. See you, Chandler. See you, Naftali. See you, Pedro. Good job.